grade 12 learners. Welcome to the life science lesson today. My name is Samantha Ragunanan. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. Just a reminder that we have a WhatsApp line available. The number for the WhatsApp line is 079 454 9535. I'll repeat that, 079 454 9535. You may send messages, but not voice notes or videos. Thank you. Let's get on with it. So today our section that we are going to cover is plant responses to the environment. And while we are looking at the examination guideline, because grade 12 learners, you know, when you start studying, I always say this, you need to start with the exam guideline. You need to look at this document that is displayed in front of you. Because without this, you will not be able to navigate that big textbook and all the notes that you've got going on there. So this section is on responding to the environment in plants. And firstly, we need to ask ourselves which paper this section is in. And this section is in paper 1 for 13 marks. Okay, now the first thing you're probably saying to yourself, it's only 13 marks. But grade 12s, that 13 marks or that 9% can be the difference between you passing or failing life sciences. It could be the difference between you getting a distinction or not. Why I like this section is because it's a very short section. Many grade 12 learners and even sometimes educators, we pass the section very quickly because it's so simple. But we don't realize the advantages of learning a section like this really, really well. Because this can be a section that can get us top marks. We can pass a section. So the content is simple. You just have to remember the guidelines that we're going to follow, okay? So let's look at what we're going to cover. We're going to cover plant hormones. But in plant hormones, there are 3 specific plant hormones that you need to know. You need to know auxins, gibberellins, and abscisic acid. So when you look at these 3 hormones, you must know the functions of each particular hormone. So at the end of the session, you should be able to recite it to me. The control of weeds using plant hormones. This is where agriculture comes into play. So if you're interested in agriculture, you'll be able to get some useful information. The role of auxins in geotropism and phototropism. This is very, very important because this is a popular exam question. They like to ask this question in many, many different ways. So if you can learn the tricks which myself and my colleague are going to teach you today, you're going to pass this section really, really well. Okay? So while you are sitting there, you should have a pen and paper and you should be making notes of all the important points. The second part of our session will be plant defense mechanisms. This is the role of the plant as a plant defense mechanism in the form of chemicals or thorns. So plants also, like how we as humans, we have a way of defending ourselves against danger, they also have a way of defending themselves against danger. So this is going to be a very, very interesting session. So stay tuned. Right, now let's get into it. Plant hormones. Now we've heard the word hormones before. You've done it before. When you did human reproduction, I communicated to you about puberty and all the different hormones that give us our female characteristics and the males their male characteristics. And we know that a hormone is a chemical messenger. What is it? It's a chemical messenger. And it takes the message from one part of an organism to the other part of an organism. So from point A, it takes it to point B. So we need to remember that the hormone isn't the part that is actually actioning the change. The target organ is the one actioning the change. So we need to understand exactly what a hormone is before we can continue to understand the functions of each specific hormone. So think of a hormone, for example, as, say your mom tells you at home, go to Grand's house and ask her to make that nice chocolate cake that she makes so well. And then you, being the messenger, you're going to go to Grand's house, to your granny's house, and you're going to say, Gran, please, can you make that chocolate cake? So who is making the chocolate cake? Are you making it? No, you are the messenger. You are carrying the message. Am I right? You are carrying the message. And who is the target organ in this case? Target organ? Your grandmother, because she's actually baking the cake. So the same way the hormones work. The hormone is a chemical messenger that transmits a message from one point of an organism to the other. Now, plants also need hormones. They need hormones because hormones help them to grow, to develop, to divide their cells, and all of those things. Okay, and hormones also help plants to action changes, because if I'm a plant and I'm feeling cold, I can't put my jacket and walk away and go say, okay, you know what, I'm going to go stand in the sunlight. No, a plant can't do that. So therefore, these hormones are absolutely useful to a plant. So we're going to unpack this in a short while. But scientists, they hesitate to call plant hormones because remember our definition of hormones. Our definition of hormones says that it is a chemical message that is produced in one part of an organism, and it is carried to another part, to the target organ, which then actions the change. But in plant hormones, it works a little differently. If we look at the note, it says your plant's hormones are also referred to as plant growth substances. Obviously, it will be plant growth substances because plant hormones help the plant to grow. That's their main purpose. Because they are referred to as plant growth substances mainly because they work in the part of the plant where they are produced. Isn't that a little different? Remember when we did hormones, we said they produced in one part and they are actioned in another. But in plant growth substances, they work in one part of the plant and they are produced also in that same part of the plant. So this makes it a little bit more interesting. So we'll move on to the next part of our work. So what exactly are plant hormones? Now, why we have this diagram in front of you, if you look at this diagram, this diagram, the heading here is, these allow plants to respond to certain stimuli in the environment. Now, what is a stimulus? A stimulus could be water, it could be light, it could be temperature, it could be all the external factors. We need to remember that plants have to deal with internal factors and the external environment. The external environment has got to do with the points I mentioned: temperature, light, water, all of those things. Those are the external factors. The hormones, they work inside the plant. Okay. So the reason that we have this diagram is to help us to remember, because in life sciences, diagrams are gold. They help us to remember things that we would not otherwise remember, because some of us, we can't learn from reading notes all the time, so we can make diagrams. So let's look at this diagram, and I want us to look at the diagram and discuss the diagram before we actually go into what are plant hormones. We can see dormancy, and we know what is dormancy. Dormancy is a state of inactiveness, isn't it? So it's almost like, when you're sleeping, you're not doing anything. There's no activities. You're sleeping, you're resting. There's no activities. So a plant dormancy is like that, where the plant is inactive. There are no activities taking place. And then we have germination, which is obviously developing from a seed. And then we have maturity as they grow further. Flowering, which is blooming. You can see the flowers there. Ripening is when the fruit turns from most likely a green color to the chromoplast color, which is red, sometimes orange, and all of those things. That's ripening. And abscission. Now, leaf abscission is basically the removal of a part of a plant, which is usually a leaf or a fruit that is no longer needed at that point in time. Now, why am I explaining this to you? You need to know what abscission is. Okay, abscission, if you look at the word itself, it almost looks like the word scissor, doesn't it? The cutting off. Okay, so let us look at exactly how plants respond to stimuli in the environment. So what do hormones do? Hormones allow the plant to bloom at the appropriate time. They allow the plant to bloom at the appropriate time. What does bloom? It's just a fancy word for flower, or to produce flowers. Okay. So what a hormone does is, remember, it's a chemical messenger. So the hormone is the messenger that sort of goes to the plant and say, hey, you can't bloom now. It's winter time. If you're going to bloom, you're going to die. So that's an important message, because even if a plant has to bloom or the flowers were to form in a plant, guess what? It would die. So what the hormone does is it waits for favorable conditions. Remember the term favorable conditions? It's when there's adequate light, when there's adequate water, the temperature is favorable, when there's pollinators. Remember, flowers need pollinators. They need bees. They need all of those factors to produce more flowers. So the hormone allows this to happen at the right time. Timing is everything when it comes to plant hormones. The next part is it allows plants to grow toward the light source. Now, I think the main part of the sentence here is light. Remember, a hormone plays an important role here, because if it must grow towards light, what is the function of light? It's the absorption of maximum sunlight for photosynthesis. In primary school, we learned it's a food making process, isn't it? Yes. So that is why hormones are so important here, because they help the plant to grow towards the light. So if the plant is not facing towards light, then it will sort of action it in a certain way, which we will discuss later on. It allows seeds to germinate at the appropriate time. So now we're talking about seeds. They need to germinate. There's a diagram of the seed germinating. Now, seeds don't have blankets. They don't have blankets to cover them when it's cold, to protect them. So there is a certain chemical that is found inside a seed that tells the seed, hey, the temperature is right now, you can germinate. Okay, so that is an important function as well. As you can see, it's a very, very interesting section. This is a section that you can remember. It's a section that you can score marks on. And I hope you're taking notes and you're working on the worksheet that has been made available to you by your teacher. Hormones induce dormancy. Induce, it means it stimulates dormant. So it makes the plant to, as we mentioned with seeds, the seeds germinate at a specific time, but prior to that, it allows the seed to remain dormant. Dormant, we said it's almost like sleeping. It's when the seed is inactive or the plant is inactive. Okay? So these are the functions of plant hormones. This is their job. This is what they do. You know, like in everyday life, people go to work, they have different jobs. A teacher teaches, an accountant works with balance sheets and all of that. This is the job of the plant hormones. So everyone has their job to do. So let's see who's everyone here. Okay? So we're talking about plant hormones. So if you look at this lovely diagram, you have the 3 plant hormones which we discussed. It's auxins. This one here, grade 12s, pay attention. I'm not circling it so many times for nothing. It is one of the most important hormones you're ever going to do. Okay, auxins. They like to ask you questions on hormones, especially auxins, because auxins has quite a lot of functions attached to it, which we're going to discuss. So when I'm discussing auxins, excuse me, when I'm discussing auxins in the next slide, I want you to pay careful attention. I want you to make notes, because they won't just ask you the functions of auxins. They will ask you experiments. They'll ask you different aspects of auxins. So you need to understand the section carefully, and you'll be able to once we're done. So that is auxins. The next one is gibberellins. So gibberellins, the spelling, grade 12s, double B, double L. Double B, double L. Remember that. Double B, double L. When you are spelling a word, make sure you say it out loud. So when you say it, that is how you must write it. Okay? So it's gibberellins. And the next one is abscisic acid. So we're going to look at these 3 in specific. What do we need to know from our exam guideline? You need to know the functions of each one. You need to know what their job is. What are they doing? So if we go to the role of auxins, there we've got some nice diagrams for you. And these diagrams are going to help you to remember. So what do auxins really do? They stimulate. Stimulate means they sort of cause that event to happen. So they stimulate cell division. So what is cell division? We're talking about mitosis. You know, remember when you did mitosis? We said that cells divide and then therefore the cell grows, they become elongated, they become larger. and all of those things. So they stimulate cell division, and this is a very, very important job, because once the cells divide, then they become differentiated. Remember, and then the plant will decide, okay, this cell is going to be a xylem. This one is going to be phloem. You know, all of those things. So the cell division is an important process which auxins do. The next one is cell elongation. So cell elongation, you can circle the word long. It's exactly what it means. It helps the plant to grow longer and longer and longer. Why? Where is it going? It's going up to absorb sunlight. Photosynthesis is your catchphrase here. Okay? And then we have the development of fruit. So when auxins are applied, for example, to the ovary of a flower, when they inject it into the ovary of the flower, that develops into a fruit, into a seedless fruit, obviously because it's unfertilized. It is able to develop into a fruit. So therefore, auxins are important in the development of fruit. As you can see here, we've got some lovely strawberries, making us hungry for some nice fruit. So that when you are thinking of auxins, think of how they promote growth in fruit. Now, when it comes to the abscission of leaves and fruit, auxins actually help to delay fruit from falling from a tree. Do you know that? It helps the plant. Say, for example, I'm a farmer and I have an orchard, and in my orchard I have apple trees. But what happens is during certain conditions, which we will come to a little later on, the fruit will start to fall. So in order for me to delay the fruit falling, I can spray some auxins, and the auxins delay the abscission of the fruit. Okay, and then the development of the adventitious root cuttings. Now, those of you who don't know, that is why we got this diagram here. That is an adventitious root system. So when you're in primary school, your teacher taught you there's a taproot, there's a fibrous root, and an adventitious root. So we remember as an adventitious root is the one that grows directly from the stem. So how does auxins help the adventitious root in stem cuttings? Now, if you look at this term here, stem cuttings. If, for example, I want to grow more rose trees. I love rose trees. I want to grow more rose trees. I take what is called a stem cutting. I cut part of the rose tree. I can't just take that stem and push it into the ground and say, okay, now you must grow and water it. It's not going to grow. But if I take that stem cutting and I put it in a solution of auxin, I put auxin powder, make a solution up, and I put the stem inside that water. Guys, you can start a business, because what's going to happen is from that stem, little roots are going to start develop, and then the roots are going to become stronger and thicker and longer, and eventually that stem cutting is, I'm going to be able to put it into the ground, and it's going to grow into a new rose tree. Okay, so you can even start that as your little hobby. So you see, life sciences is a subject that we can apply every day of our lives. Tropic movement in stems and roots. Tropic movement is basically T for turning. Basically, tropic movement means it's basically, tropism is a part of a plant, it's basically growth by a part of a plant in response to a stimulus. So if there is light, then the plant will shift towards the light. The light would be the stimulus. Okay, so this happens in both stems and in roots. So auxins help them with regard to tropic movement. We're going to discuss it a little later on. Then apical dominance. Now, before I move on to tropic movement, I want you to pay attention to this diagram here on the right hand side. Do you see how the stem is bent? Where is it going? Towards the moon? No. Remember, plants grow during the day. It's going towards the sun. It is bending. So that is a perfect example of tropic movement. So when someone asks you what is tropism, or example phototropism, you're going to, in your mind, you're going to picture this plant, and you're going to picture this plant bending towards the light, because tropism is basically, you can see it in everyday life. Every day when you're walking home from school, you can see it. Because if you take a pot plant and you put it by your kitchen window where there's a lot of sunlight, that plant is going to grow towards the light. But the next 2 weeks, if you decide, okay, you know what, I want to see if this is really working. So I label the part of the pot plant in the front, which is growing towards the light, I label it A. The part in the back, I label it B. And then I turn it around in case anyone at home gets wise ideas and tries to trick us. So the B part will be facing the front and the light. And you will see the plant is going to move again. And it's going to move towards the light again. That is what tropism is. It's absolutely interesting. Okay? Now, with regard to apical dominance. So, apical dominance, the word itself says it all. Remember, I always say to you, when you're confused about an answer, look at the question. Apical meaning coming from the apex. Where's the apex? There's it on the top of the plant. Can you see that? So what happens here is that auxins are actually produced there. So auxins are produced in that top part of the plant. And what it does is that it keeps that part of the plant growing upward. So because the auxins are on the top and they're not this side, what's going to happen? It's going to suppress the growth laterally, okay, on this side. So the apical dominance means that because of the auxins, the plant is able to go straight up for maximum sunlight. So that is the role of auxins. It's quite interesting, and it's not something that you can forget. You cannot forget this. Please don't forget this. They are everyday examples. Write down the examples so you can remember. So let's look at the role of gibberellins. Remember gibberellins? Double B, double L. We need to get our spelling right for correct biological terminology. So the thing with gibberellins is it helps stem elongation. I've already explained stem elongation. Remember when we discussed auxins? And we said that stem elongation is important for photosynthesis. If you look at this diagram here, look at this diagram. Look at these. The branches are so, the stems are so thin and they're growing upwards. So this helps for photosynthesis, but it doesn't just help for photosynthesis. Remember, there's also competition. You know, like how you have competition on your sports day, everybody wants to win and get to the to the front of the line first. So plants also, they compete with each other. And what do they compete for? No, not gold medals, not certificates. They compete for light. So if I'm a plant and next to me is a taller plant, the shade of that plant is going to block the sunlight. So what gibberellins actually do is that it helps the plant to grow upward, and it helps to outcompete the plants around it. Okay, so that aspect of stem elongation is important. It's important for root growth. Roots need to develop. So when we add gibberellins, the root becomes stronger, it becomes firmer. Remember, this is all from primary school. I'm sure you'll remember this. What is the function of the root? It anchors the plant in the soil. I can still remember this from my primary school teachers, which means they were good teachers. But the purpose of a root is to absorb water and mineral salts and to anchor. Anchor, what does it mean? So the plant don't shift from side to side. It must stay where it is, deep in the ground, like the anchor of a ship. Okay? And also the germination of seeds. Now there we see the germination of the seed. You might remember radicle, and the plumule develops from there. Okay? So it helps in the germination of seeds. It also promotes flowering. So when farmers or people want their fruits to last longer, they spray them with gibberellins. Do you know that? Because gibberellins actually, they prolong the life of a flower or a plant. So sometimes they might even add it, you know, horticulturists, they deal with flowers and the florists, you know, girls, you'll remember when you get a bouquet of flowers and things, there comes a little packet in that flowers, if you go to Pick and Pay or Spa, and in there is hormones, and that's what prolongs it. So you see, it's applicable in everyday life. So let's look at how exactly we use it in our supermarkets, because we spoke a lot about flowers. So let's talk about fruit. Everybody loves fruit. But if I go to a supermarket and I'm going to look at the fruit, I'm not going to choose these teeny tiny grapes that won't even tickle me and say hello in my mouth. I'm going to choose big juicy grapes. Am I right? So farmers know this. So what do? They spray their plants with gibberellins. And if you look on the left hand side, this one, these grapes, they don't have gibberellins. And as you can see, the size of them is much smaller. But if we look on the right hand side, these grapes, they have gibberellins. And they round and they plump and all of those things. And gibberellins don't only help the plant to develop into larger fruit, they also help increase the size of their nodes. Remember, the node is the stem. So you have your nodes, which is the little bumps, and then you have your internodes, which is the space in between there. So what they do is gibberellins help to extend it. So the nodes, so the internodes are much longer, so it gives more space for the plant to grow. So that is the exciting world of gibberellins, okay? So now we see from our first slide the importance of hormones in growth and development of a plant, okay? They help the plant quite a bit. Now, you know how we said that hormones, they help the plant to grow and develop, and they help for cell division and all of that. Not all hormones do that. We have something called abscisic acid. So what does abscisic acid do? It does almost the opposite. Let's see what it does. Now, I'm sure during autumn time or even sometimes during winter, you will see trees this color. And you probably say they look so beautiful. They're light brown and then you've got a little bit of green. But you can see what's happening here on the road. There's a lot of fallen leaves, isn't it? It's not windy, so I don't understand sometimes when we're walking home from school, there's leaves on the ground. So abscission explains why there are leaves on the ground, okay? Why these leaves are discolored, you'll be able to understand in just a moment. So the role of abscisic acid, it's an inhibitor of growth. What does an inhibitor of growth means? It means it stops or it slows down growth. So you see, not all hormones promote growth. So you must remember this word inhibit because it's part of our biological language. So we must use appropriate words, okay? So the inhibitor doesn't just mean it stops. In some cases, it slows down growth. So abscisic acid, it causes plants to become dormant in winter. So during winter, what the abscisic acid does is abscisic acid is not the bad guy here. I know I'm saying it stops and inhibits and slows down and all of that. It actually is a protective mechanism for a plant. Because imagine if a plant starts to grow in winter, the plant is going to die. So what does abscisic acid do? It removes those parts of the plants that are not needed at that specific point in time. So we remember in a plant, let's take for example a tree, it absorbs water and mineral salts. But there comes a time when there's not enough water in the atmosphere, and there's not enough rainfall, and there's not enough sunlight. So the plant has a defense mechanism, it needs to defend itself. So what does it do? Remember, your leaves, even though in primary school we call them the food-making organs, your leaves need water and they need mineral salts, and so does the whole plant. So instead of the whole plant dying, what abscisic acid does, it sends a message. And at certain points, like if you look at the point here, at this point, at the axis of the leaf, okay, there is something called an abscission zone. This is called an abscission zone. And what abscisic acid does is that it causes the leaf to be removed, as you can see, and then it becomes yellow. So it separates it. So there's not so many plant organs that need nutrients. Yes, we call them plant organs because in humans we have our liver, we have our stomach, we have our kidneys. In plants we've got leaves, okay, and other parts of the plant anatomy. So, like I said, abscisic acid also, when there's not enough water, it needs to reduce. Sometimes there are trees and there are plants that go through droughts. In South Africa, we experience a lot of areas that have droughts. So abscisic acid helps the plant to survive. What does it do? Remember, we had a structure called the stomata. And the stomata in the plant is responsible for photosynthesis and gaseous exchange, and also transpiration takes place. Okay, so if you look at the diagram here, you have the stoma is open. Remember, we call the space the stoma. So that one is open. But when the plant undergoes bad and sparse water conditions, then it closes the stoma. Okay, can you see here it's closed? So it closes it because when it's open, water can evaporate out and it can be lost through transpiration. But when the abscisic acid is released, what happens? It closes. So will water be able to leave through transpiration? No, it will not. Okay, so it's a defense mechanism. So that is the role of abscisic acid. So we also use auxins in agriculture. So auxins are used actually as herbicides. Okay, what is a herbicide? They are hormone weed killers. So in the herbicides, like if you go to, in South Africa, we have stores like Builders, and we've got nurseries and all of those places that sell plants. So they also sell weed killers. Traditionally, what people would do to get rid of weeds, I'm sure you might even be doing this at home, where you've got the weeds growing in between the paving, and your parents tell you not pull the weeds out, or you have to go in the garden. Your grandmother, maybe you go visit her, pull the weeds out of the garden because they're getting in the way of the cabbage, all of those things. So auxins help us with this, because what auxins do is they actually cause the weeds to overgrow so that they end up dying. The main factor that controls weed growth in these herbicides are auxins. These herbicides can only kill weeds. So that means I can put this herbicide next to my rose trees. It's not going to kill my rose tree. Many people think that when you use herbicides, they will kill your other plants. They won't kill your other plants. They are made specifically to kill weeds in this case. What is the advantages of hormone weed killers? They are non-toxic to animals and humans, which is really good. So it won't be poisonous. There is no longer a need for weeding the garden. This is so tiring. So all you do is you go spray your auxins on the weeds and they will kill the weeds. These at the bottom are examples of types of auxins that are used as weed killers. Let us move on. So remember in the beginning on our exam guideline, we said that so thus far we know the functions of auxins, of gibberellins, and of abscisic acid. We know what they need to do. But part of our exam guideline says that we as grade 12 learners need to know two things. We need to know the role of auxins in phototropism and geotropism. Phototropism and geotropism. We need to know these terms. We need to know what it means. And most likely, they're not going to ask you the definition. This question comes out in diagrams all the time. Last year, there was an investigative question. You will see when my colleague comes after the break, it's a very, very important section. So even when we are doing the examples, you need to pay attention, because we do the content in these sessions, but we also do past year papers and examples, because they don't ask you like how it is in your textbook. They ask you in different ways. And an investigative question is compulsory in every life science exam. It must be there, even in a test. And this section, they like to ask a lot of experiments. So if by now you don't know what is the control of an experiment, you don't know what is a dependent variable or independent variable, this is the section you're going to find it in. So you need to brush up on that. Okay? So let's talk about the role of auxins. Auxins is responsible for phototropism. So obviously that would be movement of the plant towards a stimulus, which in this case is called light. Therefore, photo meaning light. Tropism meaning turning towards. So if there's a light shining to me, I will turn towards it. That is phototropism. Okay? So let us look at these diagrams. Now you need to know what is going on in these diagrams. So you must please pay attention. Now remember we spoke about apical dominance. We said that auxins are produced on the apex of the stem. Okay, so auxins are made there, and you can see the light. Where is the light coming from? It's coming from the top. Now, what we need to know about light and auxins, they don't, even though auxins cause plants to move towards the light actually kills auxins. So, what's happening here? There we see the auxins. They are accumulating. They are being spread out in the stem. They are moving downward. What is causing them to move down? The pull of gravity. That's what's keeping us on the ground, isn't it? The pull of gravity. That is why we're not floating around, because of the force of gravity. Even though we are not geography learners, in this section we need to know what gravity is. Okay? It's the force of attraction of the earth on a body. So let us look what it says. The auxin moves down the stem. The auxin diffuses down the shoot, stimulating growth. So there's the auxin moving down. So they say the auxin diffuses. Now remember, in your stem, we got xylem and we got phloem. So the auxin is actually diffusing through those substances, through the phloem and into the stem, and causing the stem elongation. So in the next diagram, now here the difference between the two diagrams is, if you look at where the light is, in this diagram the light is coming from directly above. In the second diagram, let's call this diagram 2, the light is coming from this side. So when the light is coming from one side only, in life sciences we've got a fancy term, we call it unilateral light. Uni meaning one, lateral meaning from the side. So here's the light coming from one direction, our unilateral light, and it's approaching the stem at an angle. Okay, like that. So if one side of the shoot is in the light, the auxins diffuses away from the light. Can you see? On this side, there is nothing. You won't find auxins there, because we know what happens when auxins are exposed to light. They are killed by the light. So the auxins diffuse. They keep moving, keep moving, and they are now on the one side. Can you see where the auxins are concentrating? On the left side of the shoot. And what happens here? If they are still concentrated on the left side of the shoot, on this side of the shoot, because it has more auxins, it grows faster. So this side is going to grow faster. Why is it going to grow faster? Remember, auxins are responsible for cell elongation. It's responsible for making cells longer. So even though this side is going to grow normally, the side which has the auxins, which is on the far side, it's going to bend, because what's happening? Cell elongation is occurring. And where is it bending towards? Towards the light. So if you read the caption, just to round up there, the side of the shoot in the shade has more auxin. We know why it has more auxin, because the auxin is killed by the light. So it grows faster, causing the shoot to bend towards the light. There's it growing faster, and there's the shoot bending towards the light. Okay, so that is a role of phototropism, a role of auxins in phototropism. So when you look at a diagram, you first need to, in the exam they will either give you a diagram of a root or a stem. You first need to determine, is there light involved? You can't talk about light if the question doesn't show you that there is light in the question. When I go to the next slide and we discuss geotropism, you must look at the question and see, what are they talking about? Is there light involved? Is there gravity involved? Because these are the two factors. Now, before I move on to the root, I need to tell you that auxins are great. They're great in the stem. What they do is they promote growth. But they do the opposite in a root. So when you find auxins in the root, it inhibits growth. That is something you must write down right now. Auxins promote growth in the stem. It inhibits growth in the root. Many learners do not understand that very, very important point. Therefore, they get the experiments wrong. Therefore, when they are responding to a question, they become confused. Because they think, but you're not auxins. The moment they see auxins, they say, oh, growth, cell division, cell elongation. Not really. In the root, it inhibits growth. So if you still don't understand it, I'm going to explain to you now how it actually inhibits growth. So if you look at this, okay, you're probably wondering, okay, here we have, okay, this is geotropism. So this is when there is growth that occurs towards a stimulus, and the stimulus is what? Gravity. Geo meaning, geo coming from the word gravity, okay? Tropism. So what does this tell you already in the name? In plants, in the stem, what happened? We had phototropism, and we said that the plant is growing towards the light. So what do you think geotropism is? Is when the root grows toward gravity, isn't it? Toward the ground. The ground is pulling the root down. You need to understand the term. You need to know what the term means, because you're going to come to positive and negative. So for example, if we go back, let's look at this phototropism. Now there's two types of phototropism. You can have positive phototropism and negative phototropism. So what is positive? Positive phototropism is when the plant is growing toward the light. That is positive phototropism. But if you look at the root, where's the root? The plant is the stem is growing upwards. So that's positive phototropism. Where's the root? The root is not going upward. The root is going downward. Can you see that? The root doesn't need to grow towards the light. The root is not interested in the light. The root is interested in water and mineral source. That's what it wants. So it will be negatively phototropic. Okay? It's obvious now. Stem needs light. Stem is positively phototropic. Root needs water and mineral salts. It's not going to find it up there. It's going to go down. So it's negatively phototropic. Okay? Okay, so now we're going to be able to understand geotropism a lot. Similarly. Now, from our knowledge now, from the slide before, what do you think the stem is going to be? Because if the root is growing towards gravity, and we're talking about geotropism, so the root is going to be positively geotropic. Because why do we say positive? Because it's growing towards the stimulus. What is the stimulus? It's gravity. Okay? And the stem, what about the stem? Is the stem positively geotropic? No, because the stimulus here is not light. The stimulus here is gravity. So the stem is going to be negatively geotropic. We need to understand the terms. If we do not understand the terms, phototropic, geotropic, positively phototropic, negatively geotropic, how are we going to understand the experiments? We're not. So terminology in life science is important. When your teachers are giving you those terminology tests, you should be paying attention. So after the section, I would expect you to write down all the terms that we've discussed here in this section, and try and find the meanings and learn the meanings. Now remember, this section you can study with diagrams. So it's not like I'm telling you to go learn a whole lot of content. I'm just telling you, learn with diagrams. Okay? So let's look at what happens here. Now here we got a plant is laid down on its side. I'm reading this part here. If a plant is laid down on its side, oxygen gathers in the lower half of the stem and the root. Why would the plant lay down on the side? Basically, sometimes what happens, erosion occurs, there could be rocks in the way, but the plant ends up laying on the side. Anything can happen in nature, okay? We know that. But in any case, we ended up here with a plant which is laying on its side. And why is the oxygen gathering here at the bottom? Remember, we are studying geotropism. Do they mention anything about light here? They don't mention anything about light. So you should not be talking about light. Here we talk gravity and we talk about growth. Okay, so very quickly, if a plant is laid on its side, oxygen gathers in the lower half of the stem and the root. We're going to go very quickly because we've discussed the principles already about this, okay? So the force of gravity, it pulls on the oxygens, and the oxygens gather at the bottom. And what happens as a result? The root continues to grow downward. But because the oxygens are gathered here, Okay, the auxin slows down growth in the root, so the root curves downwards. Remember, we said auxins has the opposite effect. So even if there is auxins here, we would expect it to grow, but it doesn't cause the plant to elongate because it's in the root. It inhibits the growth. But it doesn't stop the growth, because if you look at the top of the root, these cells are still normal, so they still continue to grow. And as a result, the auxin stimulates growth in the shoot, and then the stem curves upward. Okay, so that's just a summary of what we've already discussed. It's in a diagram. So you should be able to compare. I'm not going to do both sides of the table. We will just do one side of the table, because obviously it will be the opposite on the other side. So let's look at phototropism. It's produced at the stem and the shoot. It moves downward. There is even distribution because of the pull of gravity, and also remember, the auxins are diffusing into the phloem. And it grows, the stem grows upward because it causes, auxins cause cell elongation. Unilateral light is a single light from a single direction, and the auxins concentration will be high, and then we know that they stimulate growth and it is bent towards the light. So this is not a comparison. You're seeing this table in front of you. It's not a comparison. It is a summary of what is the role of auxins in phototropism and what is the role of auxins in geotropism. Okay? And then you could get an experiment with a clinostat. Now, a clinostat is an apparatus which you will find in a lot of experiments. Basically, it's a disc. And on this disc, it rotates. It moves round and round. And sometimes if you put a plant on it, and you put the plant in a box like it is in the diagram, it fools the plant into thinking there's light all around, but there's no light all around. Let's look at the diagram. I got a little tip for you. Most of the time, the diagram with the clinostat where it is rotating is usually the control. Okay? So let's look at it. If you look at this experiment, here there is no clinostat, and the plant is exposed to light only on the one side, and the stem grows towards the light. There we go. The plant is growing towards the light. The light is supposed to be exposed there. But here we got the clinostat. And what happens with the clinostat, even though the light is only coming from one end, the plant is fooled into thinking, because it's rotating, it's fooled into thinking that there is light it's exposed to light from all sides. Okay? And it causes the plant to grow straight. Right, and then we look, and there's another further example of a clinostat. Just an example of the type of questions you can get. You will find these questions in give the correct biological terminology. And we're not going to go through all of them in the interest of time, but we are going to expose you to questions and tell you where you can find these questions. So you will get them as biological terminology. The movement of roots downwards would be positive. Remember, positive geotropism. The growth response of plants to the stimulus of light, it's phototropism. And the main hormone controlling leaf abscission, obviously it will be abscisic acid. The apparatus which is used, you must know this, for demonstrating geotropic growth, it is the clinostat. Okay, so we are going to take a short break. But please don't go anywhere. My colleague, Ms. Shabdam Suknanan, is going to be taking you through a very, very important part of plant response to the environment. Okay, so stay tuned, take a short break, and we'll be back in a minute.
12 learners, welcome back to the second half of the grade 12 lesson based on plant hormones. Learners, what we're going to be doing now is very important as it relates to all the content of plant hormones. We are now going to apply it to our investigative questions. Yes, investigative questions are really tormenting for you, but I promise you, if you know a few terms that you can go through and revise all the time, you will get used to your investigative questions. So now let's look at the second half. We are now going to be looking at plant defense mechanisms. A very simple part which can be easily scored by you, right? It simply means how actually are plants defending themselves, just like animals can defend themselves with their hands, with their nails. Plants also have defense mechanisms such as your thorns, right? Which is a mechanical defense mechanism, as well as your chemicals. So if you look at here, it's a chemical defense. Most plants actually give off an odor or a smell, or they can even taste very bitter. And should an animal eat these plants, you'll find that the animals themselves will be able to, right? You'll find here.
You have been given power to decide whether you want good results in your life, in your schoolwork, or you want bad results. As Tony Robbins said, it is in the moment of your decision that your destiny is shaped. And your destiny is shaped right now with the decisions that you are currently making in your schoolwork. So decide whether you want to be an A student and start working on it, because there is power inside of you. The infinite power is inside of you, and the time you decide to tap in that power, you will become who you want to become in your schoolwork. Believe in yourself. Believe a little more in yourself, because to those that believe, nothing is impossible. You have done this before. You have passed your tests before. You have went into a higher grade before. It is possible because you have done it before. Do not doubt yourself. Keep believing in yourself. Keep pushing, because this can be done and you can be great.
Welcome back, learners. Sorry for that technical error. We just go back to looking at plant defense mechanisms. First of all, you need to know that plants have adapted to prevent herbivores from eating them, and that is by means of a chemical mechanism whereby the leaves can turn bitter, right? And you also have, for example, another defense mechanism whereby you have thorns, like your prickles on a rose bush, the thorns of an acacia tree, the spines of a cactus. So these are mechanisms that prevent the animals from eating them. And why do they need to do that? So if every animal eats every single plant, plants can become extinct. Therefore, plants need to defend themselves by chemical means and by mechanical means such as thorns. So this you can get for 2 marks, that chemicals and thorns are the 2 mechanisms of plants defending themselves. Now comes the tricky part, and that is all your investigative questions. So we're going to look carefully at the investigative question, analyze it, and then interpret it. Okay, so let us read. We're going to still look at the role of auxins. A group of learners conducted an investigation to determine the effect of auxins on the growth of stems in bean seedlings. Right, learners? So we're going to see here, we're going to look at here that there are 30 bean seedlings that were allowed to germinate for 5 days to produce seedlings. So let's look at it. There were 30 bean seeds. Right. Now they took the 30 bean seeds and they allowed them to germinate. And once they germinated and they produced a young stem and a young root, which is called a coleoptile, right? They took the 30 bean seedlings and they divided them into 3 groups, A, B, C, of 10 seedlings each. All right. So now, once they looked at the seedlings, here you can see group A, group B, and group C. These 3 groups were treated differently. Right, so let's see, the procedure was as follows. 30 bean seeds were allowed to germinate for 5 days. Okay, so not 1 day, 5 days. It gave it time for every bean seedling to germinate. Once they germinated, the seedlings were divided into 3 groups, A, B, and C, of 10 seedlings each. The tips of all the seedlings were cut at the same length. Now, learners, we need to look at this investigative question and see why was 30 bean seeds used and not like 5. When it's more bean seedlings, it means it increases the sample size, which means it makes that investigation reliable. Then you look at it and you say that each group had 10 seedlings each. That means each group was equally divided, kept the same amount of seedling. That is now validity. So in experiments, you look at reliability and validity. Validity means you must keep things the same to ensure there is fairness in that investigation. And what were the first 2 things that we learned about? We said that each one was divided into 3 groups, right? But in each group there were 10, 10, 10. Okay? And then you find now all of them were cut at the same length. So the same length of the stem was cut. That same means validity. Let us look now at group A. What was done to group A's little plumule? So you find that that coleoptile growing on the top is a plumule. It develops into a young stem, and the one that's growing down is the young root. Okay, so now we are looking at what? Determine the effect of auxins on the growth of stems in bean seedlings. On the growth of stems. Right, so now you can see in group A, the tip was cut. So they took off the tip. What did we say earlier? That auxins are found in the tips of roots and in the tips of stems. Remember that. Where are auxins found? Tips of roots and tips of stems. So if in the first group A, all the tips were cut, we are actually removing the auxins. And then once we removed it, then they replaced it. So that is group A. Okay, now let us look at group B. In group B, you find now, in group A, here's it, the tip was cut and replaced back onto the top of the stem. Now we're going to see in group B exactly what happens. So in group B, you can see that here they removed the stem tip and never replaced it. So what happened? We have removed the auxins. We've removed the auxins completely. So there's the difference. A stem tip was cut, then replaced. B, stem tip was cut completely, not replaced. Which means now B, in group B, all the seedlings there, all the little plumules will have no auxins. Right, and let's look at how was group C treated. In group C, they cut the piece of the stem tip. The stem tip was cut here, right? Once it was cut, it was removed. So in group C, a piece of the plastic was placed on top of the cut surface, and the cut tip was then placed on top of the plastic. So here is your stem. It was cut. Then at the cut part, they put a piece of plastic, and on top of the plastic, they put the cut tip, which means that the tip containing auxins was now placed on a piece of plastic and then the rest of the stem. That means that the tip containing auxins was separated from the rest of the stem. Now, before we look further, let us analyze this diagram. Right? Now we know they were treated differently. We can see which one becomes my experiment. My experiment, remember learners, experiment is always the one with the factor that is being investigated. And what is the factor here? We want to see the effect of auxins on stem growth. Effect of auxins on stem growth, right? So now this one has your auxins. So that becomes my experiment. This one, group B, has no auxins. That becomes my control. And eventually we need to compare the results to see does really auxins have an effect? And why must we put group C? We want to see now, if we cut the stem tip and if there is any growth in group C, it means auxins can diffuse through plastic. But if there's no growth in group C, it means the auxins were at the tip, but they couldn't move down to bring about stem growth. So that is how you need to apply your knowledge to investigative questions. Now let's look further. Same diagram. The seedlings in all the groups were placed in a dark cupboard for a week. The seedlings in all the group were placed in a dark cupboard for a week. Now, you've learned earlier that auxins are sensitive to light. Now, why will we put auxins or plant seedlings in a dark cupboard? I want you to remember one thing, that auxins are sensitive to light and they're also sensitive to gravity. So, because they found in the stem, which is subjected to light, and remember they found in root, subjected to gravity. So, the auxins react towards light and they also react towards gravity. So, even if there's no light, auxins can still react towards gravity, depending on the position of the plant. Right, now here, the seedlings in all the groups were placed in a dark cupboard for a week. So automatically, you cannot say that they were growing towards phototropism, they were growing towards light, or it's phototropism. There is no phototropism here because there is no light. So they were placed in a dark cupboard. So we wanted to eliminate the factor of phototropism. We wanted to eliminate light. The growth of the stem was then observed. Why? What factor are we actually concentrating on? We want to find out the effect of auxins on the growth of stem, not the effect of light on the growth of stem, or not the effect of gravity. What are we looking for? The effect of auxins, because that's how the three groups were treated. Right, now we want to look at the growth of the stems. So now children, ask yourself, if you're the investigator, what did you control? You control how you treated the three groups. You cut the stem and tip off, then you replaced it. The stem tip was cut off, which means you remove the auxins. The third one, you cut the stem tip and then you place it on the plastic, which means now auxins cannot move downwards. Always remember of the behavior of auxins. Auxins move always from the tip, they move down and then they bring the reaction up. Okay, so what is going to be our independent and dependent variable? These are two terminologies you need to know for investigative questions. One is validity, one is reliability, one is independent variables, and your dependent variables. Now let us look at this experiment here, or this investigation. What are you able to control? You are able to control the effect of auxins. So that becomes my independent variable. But what can't you control? What you still need to measure? You need to measure the growth of the stem. And if the stem grows, you know that auxin has its effect. If the stem did not grow, you know there was no effect. So what you can't control and you still need to determine becomes our dependent variable. So in this case, effect of auxin becomes independent and the stem growth becomes our dependent variable. So now let us look at the questions involving this investigation. Identify the dependent variable in this investigation. Dependent means, what does the growth of the stem depend on? It depends on the effect of auxins. So dependent, my growth of the stem depends on. So automatically our independent variable will be stem growth. Okay. Why did the learners cut the tips of the young stems? Why did they cut it? Because they want to know whether auxins have an effect on stem growth. So they want to find out whether auxins are going to be causing the stem growth, because auxins are found where? In the stem tip, and they cause stem growth. So we cut it because we want to remove the auxins, because the auxins are found in the stem tip, because that's why we want to know, is there an effect? If we remove it, let's see if the stem grows. If we leave it, let's see if the stem grows. Therefore, they cut the stem tips because we are now carrying out an experiment. We're carrying out an investigation. Okay, give one reason why 10 bean seedlings were used in each group. 10, 10, 10. Not 10, 8 or 12. We kept them constant, right? Because we want to see what? Validity. Okay, so give to increase reliability or validity of we want the same results, reliability. We want to make it fair, validity. Okay, so you can say to increase reliability and for validity, because we're keeping it the same. We want the same results. We want to compare the same things. We want to make sure it's fair. So therefore, validity, fairness, reliability, we want to get the same results. Okay, we don't want a factor to be affected, and that another factor must now cause a disturbance in the results. So remember, whenever the size sample is bigger, we talk about reliability. If we keep it the same, we talk about validity. Right, write down the letters A, B, or C of the groups where there will be no upward growth of plants. So learners, remember that in A, the stem tip was cut with auxins and replaced. So nothing was separating the stem tip now. Just that it was cut and placed back, which means auxins moved down in A. And in B, remember it was just cut off. There was no stem tip, no auxins. And remember in C, it was cut and replaced, and there was a plastic that was separating the stem tip from the rest of the stem. So I would not expect growth where. I wouldn't expect growth in B because the stem tip was cut, auxins were removed. B and C, I know that auxins move down, further down, and then cause upward growth. So I wouldn't expect any stem growth in B and C. But let us look at the answer. There will be no upward growth of the stem in B and C. Right, describe how auxins cause apical dominance. Our first presenter spoke to you about apical dominance. And I want you to know this because there is no question paper that will not ask you about apical dominance relating to plant hormones. Now, apical means apex. Apex means tip. Dominant means that this auxin is dominant at the tip. It's dominant. It dominates. Okay? It's right at the tip. It's at the apex, and it dominates how? It only causes the plant to move upwards. It doesn't allow it to grow sideways and become more bushy. Right. So therefore, how do auxins control apical dominance? They allow upward growth, and as long as auxins are found in the tip, the plant will just grow tall and it will hardly have any side branches. Okay. So let us see. The presence of auxins at the tip, as long as auxins are found at the tip, they'll dominate the tip and they will cause upward movement of growth. Right? And as long as they're there, they are the apex, they call it apical growth, then they will prevent side branches. We call side branches lateral branches. Do not say lateral growth. Lateral growth means you're becoming fatter. It stops the forming of lateral branches, not lateral growth. Okay, so it allows apical growth, the plant to become taller, but as long as auxins are there, it will prevent lateral branches from forming. So the plant will have less side branches. Right, so the presence of auxins in the stem tip stimulates upward growth and inhibit the development of lateral branches. Next question, name the plant hormone other than auxins that promote the germination of seeds. So besides auxins now, what other hormone did you learn about earlier? Gibberellins. G for gibberellins, G for germination. So gibberellins are associated with the germination of seeds. So as soon as spring arrives, the farmers need to soak their seeds now, just before the rains can come, hopefully. They soak the seeds in gibberellin solution. So the minute they plant them into the ground, all the mealies and the wheat and the rains come, and then quickly they need to start germinating. They do that with cabbages as well, because cabbage and onions and tomatoes are staple foods of the population today. And therefore, even the potato seeds, that they've gone expensive. Expensive, why? Because these are no ordinary seeds. The seeds have been soaked by gibberellin solution to ensure that they germinate fast, because we cannot wait for the rains, because we need food for the growing nation. It also helps in defending food security. So learners, the auxins, they will actually help for stem growth, but the gibberellins help for the germination of seeds. Here's your gibberellins, germination of seeds. Again, name the plant hormone that inhibits. Please remember, the word inhibit means stops. It's the opposite of promotes. It's the opposite of stimulate. So if you get stimulate, it means allows and promotes. But if you stop it, you inhibit it. Right. So name the plant hormone that inhibits the germination of seeds and brings about dormancy in seeds. Now, you don't want your seeds to start germinating now, because if it germinates now in May, there's no rain. So we want those seeds to stay dormant. Dormant means inactive for a while. So what do? If you need to actually transport seeds from South Africa to Europe, because in Europe there's no mealies. Now they want mealie seeds. So you want your mealies seeds to go in the aeroplane, but they must not germinate. So you simply spray what? What causes seed dormancy and what prevents the germination of seed? Gibberellins promote germination. What prevents the seeds from germination? It's abscisic acid. Let us see if we're correct. It's abscisic acid, okay? So abscisic acid is associated with the dormancy of seeds. Now we go to another investigative question, learners. And this one, take your pencil and underline. What is this investigation doing? Ask yourself, to determine the effect of plant hormones on the plant growth. Again, now you're looking at the effect of plant hormones on plant growth. So similar thing. We want to see the hormones effect on plant growth. Now the procedure was as follows. So this is now another form of a procedure. Okay. So picture what is happening. Two pot plants, 1 and 2, of the same species and age were used. What is significant about that first sentence? Same species and age. It means they're keeping the experiment fair. 2 pot plants, same species, not a rose and a daisy. 2 rose pot plants. 2 pot plants with little rose trees, okay? Same age, not one's older, one's younger. So therefore it's validity, it's fair. That's the first sentence. And there's only 2 pot plants, 1 and 2. Then the apical buds of both plants were cut at the same length. What is an apical bud? An apical bud is a bud at the tip of the stem. A lateral bud is a bud on the side branches. So they are cutting all the apical buds, like what you will do if you have a gardener and he comes and prunes off your trees. When he's pruning and cutting all the apical buds of the rose trees, he's removing the apical buds. He's removing the auxins. So the apical buds of both plants were cut at the same length, not one short, one long. They were all cut at the same length, okay? So that is also what? Validity. They're cutting it at the same length. The cut surface of plant 1, so now in pot plant 1, when he cut the apical bud, the top of the cut part, the surface, was placed with petroleum jelly. You all know what's petroleum jelly? Simple Vaseline. Okay? So when he cut off the stem tip and removed the auxins, he simply placed Vaseline. And you know, Vaseline does not allow anything to diffuse through. It's like fat. Okay? Nothing can go through. It's like oily. So it won't go through. So there, nothing, no water or anything can go into the cut tip. So therefore the auxins were removed and nothing from the air can go through into the cut part of the stem. The cut apical bud of pot plant 2 was placed on a block of agar for 2 hours. So remember, in pot plant 1, the cut tip was removed, thrown away and sealed with Vaseline. In pot plant 2, the stem tip was cut. Then the cut part, which contains auxins now, was placed on a block of agar. Agar is actually jelly. Okay, learners? Small piece of jelly, and you put the stem tip on there. Here is your agar block. Right. Now, here's your agar. So what happened is, now remember, the stem is cut. Now you took the cut tip, placed it on agar. In jelly, agar can diffuse. So agar can only diffuse into jelly, not into Vaseline. So it can diffuse. Then they took this block of agar and they're going to replace it on top of the cut tip, cut part of the surface, right? We're going to look at the diagram just now. The block of agar jelly, so see, look again, the cut apical bud of pot plant 2 was placed on a block of agar for 2 hours, giving it enough time for all the auxins from this cut stem tip to diffuse into the agar. For 2 hours they left it. Then they took only the agar, not the stem tip. because all the auxins are emptied now. They went into the agar. So they took the agar block and placed it on the cut surface of plant 2. Okay, both plants were exposed to the same environmental conditions for 2 weeks, not 3 weeks one and 2 weeks one, same amount of time. That's validity. Same environmental conditions, like same temperature, same amount of water was given, same soil was given. Okay, the growth of both plants were observed at the end of this period. So the growth of both plants was observed. Here is your picture now of what you read. Now we're putting it in the form of a diagram. Okay, now here is pot plant 1. Can you see, dear learners? Here, remember that we now remove this cut tip. We cut it, the tip, we threw it away. So now auxins are no longer dominant there. They're out. So when auxins are no longer there, they're no longer going to cause upward growth. Can you see what's happening here? Because there's no auxins, we are now allowing for lateral branches. Can you see? So the auxins are removed. There's no apical dominance now. And what? There's no more upward growth. Because the auxins are removed, they are causing now side branches, lateral branches. And remember, in pot plant 2, learners, this here is actually part of the entire agar. Okay, don't get confused. This is the entire agar block. All right, here is the agar block. Okay, so this is the agar block. You can see here that here the stem is growing, and now you find that the auxins which are in the agar is going to cause the auxins to move down and cause upward growth. Look at the difference here in the length. This one's moving up. Okay, this is moving up. That means now the presence of auxins from the agar went down, causing apical growth. And here, because there's no auxins, it's causing lateral branches. Right, so explain why the stem in pot plant 2 grew upwards. It's because in plant 2 you can see it grew upwards because the auxins moved down and caused cell elongation up. All right, and here state why the apical bud was placed on a block of agar jelly, as we said, so the auxins can move from the apical bud into the block of agar jelly. Okay, describe the results obtained for plant 1. The result is that in 1, the stem tip was cut, auxins were removed, and because the auxins were removed, the stem tip stopped growing upwards and allowed for lateral branches. Okay, explain how fruit farmers can use the knowledge from the results in question 2. So how can fruit farmers do? They prune their fruit trees, they prune all the peach trees, and by pruning it, they're not allowing the peach tree to grow up. It's allowing for more side branches so you can get more fruit, and more fruit can be harvested for the economy as well. Lateral branches will develop because you're removing the auxins, you're cutting the stem tips, you're removing auxins, no apical growth, more lateral branches, more fruit will increase, you'll have more shorter trees with more bushy trees, more lateral branches, and more fruits will form. The third one now. This is another investigation, learners. And this investigation is to determine the effect of auxins on the direction of the growth stem. Now we're talking about direction, not just growth. The procedure, again picture it. 2 plants, 1 and 2 of the same species and age group. What we said, validity. Apical buds of both plants were removed by cutting at the same length. Again, same length, validity. Same species, age, validity. Only 2 pot plants. Again, the cut apical buds of both plants were placed on separate blocks of agar for 2 hours. Let us now see how exactly was this. 1 block of agar was placed on the right hand side of the cut surface. So here's the cut surface. 1 was placed on the right hand side, right? And the other block of jelly was then placed on in plant 2. In plant 2, it was placed on the left hand corner. Okay. Both plants were kept in the dark box for 2 weeks because we don't want light now. Okay, we are not talking about light. We just want to see what the effect of auxins on the direction. Here there's no light. Both plants were kept in the dark. We want to eliminate light. The direction of the growth of the stem of both plants was observed. So let us see. Here is the actual experiment. Here's pot plant 1, pot plant 2. both the cut surfaces were removed. Okay, the cut parts were removed, placed on agar, auxins diffused into the agar, and then how was it different? They put the block of agar here on the right hand side of this cut tip. They put this agar block on the left hand side of the cut tip. Right. Now let us analyze what's going to happen here. We know there's no light, okay? So auxins always are going to move from the agar down. Auxins are going to move from the agar down. So here there'll be high concentration of auxins on your left hand side. And here there'll be high concentration of auxins on your right hand side of this pot plant, okay? On the right hand side of this pot plant. Remember, your left becomes the pot plant's right and your right becomes the pot plant's left. So although this is my right hand, but on the pot plant side, if you turn around, that's the left. So they put it on either side. Right, now we're going to see. Give a reason why the apical buds were removed. We're removing them because we want to remove the auxins, because auxins cause what? Apical growth. You see? To remove the auxins produced at the apical buds. Why were the auxins placed on the block of jelly? To allow the auxins to diffuse into the block of jelly. We wanted not the stem tip, but just the auxins to move down. Right. Now they treated pot plant 2 differently. Pot plant 2 now, in the box, here's your pot plant. Remember, that's your plant and it had the agar block here. So pot plant 2 was exposed to unilateral light. We said light coming from one side. So the light came from the left hand side by removing the lid. So they actually now remove the lid over here, right? And they allowed light to come through here. So they cut it and allowed light to come through on this side. The light was coming through there. Explain the results that we obtained. Now we treated this one differently. We are now taking the lid off the box and allowing light to come from one side, from the left. What did you learn, learners? Auxins are sensitive to light. They always move to the shady part. So they're going to move from the part where there's light to this part here. From the left hand side, they're going to move to the right hand side. And remember, a high concentration of auxins in a stem. Remember, we're looking at the stem. High concentration of auxins in a stem will promote cell elongation on that side. So this stem now, because there's more auxins here, there's going to be cell elongation here. It's going to elongate faster and faster and faster. And then the stem will bend in the direction of the light on the left hand side. So the stem will bend towards the left. They move, light is coming from the left, the auxins move to the right on the shaded part. And then high concentration of auxins in the shaded part will promote cell elongation in the shaded part and cause the stem to bend towards the left, towards the light. This is called now phototropism. Okay. So let us see here again, children. The auxins will move from the left hand side. Okay. It's going to be here now. Right, we're just going to go back here to the
Believe
to the
in yourself.
Believe
a little more in yourself. Because to those that believe, nothing is impossible. You have done this before. You have passed your tests before. You have went into a higher grade before. It is possible because you have done it before. Do not doubt yourself. Keep believing in yourself. Keep pushing because this can be done and you can be great. The road to success is to commit to yourself. In order for you to become who you want to become as a student, you need to commit to reading your books, to studying, to bettering yourself. I know it's not going to be easy. Actually, no one said it's going to be easy. But if you put in the work, the results will show in your life. Tap into your power. Sometimes you will feel like putting away your books, and not reading as much as you're supposed to be. But I want you to commit, because through commitment, there is infinite power.
Okay, welcome back again, learners. So we're going to see that auxins move from the light part to the shaded part, cause cell elongation on the side of the shade, and then it will elongate, and then you find your auxins, the stem will bend towards your light. All right. And then we're going to say, explain why rose farmers now and again remove the apical buds to prune the rose trees, because they want more roses, more side branches for more roses. Okay, removing the apical buds means preventing apical growth and allowing more lateral branches. Lateral branches means we're going to get more roses. And you can see more lateral branches increase the yield of more roses, and you find it will be easier to harvest because now you can pick up the roses from your side branches, and not if it's very, very tall, then you need ladders to now go look for roses. Here, if it's on the side branches, it's easy to harvest. State one reason why the apparatus was placed in a dark cupboard for the duration of the experiment, because we don't want to see the effect of light now on auxins. We want to see the effect of actual auxins. What do? Do they cause stem growth, or must they only have light? But we can see that auxins can cause stem growth. As long as auxins are present on the stem tip, the plant stem will grow. Right. To eliminate the effect of light, that's why we place it in the dark cupboard. And we don't want phototropism to affect the results. We want to see the effect of auxins. Right. Now another experiment here is a bean seedling, learners. So this bean seedling, a seedling was placed horizontally. Horizontally means lying flat in the dark, so there's no light, as shown in diagram A. So here's your bean seedling. Remember that there's two parts, a plumule and a radicle. Now how will you know which is the plumule? Remember, plumule always forms the stem, radicals always grow down to form the root. You can see here the stimulus is the gravitational force. So a seedling was placed horizontally, not vertically, not upright, in the dark, as shown in diagram A. Diagram B shows the result after 5 days. So remember, diagram B is not another treatment. Diagram B shows the results of diagram A. You can see that all these dots at the bottom here are what? These are your auxins. Okay. How come the auxins went down? Because gravity pulls the auxins down. So this is the force of gravity that's pulling the auxin on the lower side. Now in a root, remember here's the root. Now it's formed, there's the root bending. Okay. So in a root, high concentration of auxins in a root will not cause cell elongation on that side. In a stem where the auxins moved on the high concentration on the shaded side, it caused cell elongation there and uneven bending, unequal growth, and the stem bent. But now here also there's unequal growth, because here in a root, please learners remember, in a root, high concentration of auxins will inhibit cell elongation. Okay, so high concentration of auxins in a root inhibits cell elongation on that side, and on the opposite side where there's less auxins, that part will start elongating more and more and more and more, and then bend towards gravity. So remember this. That's all you need to know. That's important and tricky. Lots of auxins in a stem on one side, that's where cell elongation will occur, on that side, in a stem. High concentration of auxins in a root on that side, no cell elongation, but cell elongation will occur on the opposite side, like that. Okay, and it will bend towards the stimulus. In this case, it is gravity. Give the term for the movement of part of a plant in response to a stimulus. They're not talking of light, they're not talking of gravity. So just the movement of part of a plant towards the stimulus, it's just called tropism. But if it's towards gravity, geotropism. If it was towards light, phototropism. Before, it used to be towards water, it was called hydrotropism, but that's no longer in the syllabus. Which plant hormone stimulates the growth movement? Always. Movement towards gravity, movement towards light is only auxins. Give any other two functions of hormones mentioned in question 2. And the functions are, what do auxins do? It brings about the bending of plants, right? It brings about tropic movements. That means movement of the root towards gravity, movement of the stem towards light. It brings about cell division, so the plants can, more cells, more growth. It causes the plants to elongate, so the plants can grow tall. Responsible for apical dominance. They dominate the apex, and as long as auxins are found in the apex, there'll be less lateral branches. The moment the auxins are removed, there'll be more lateral branches. This is called apical dominance. And also auxins cause roots to develop so if you take a stem cutting and you put auxins in there, yes, roots will develop as well. Right. Does the accumulation of the hormone auxin inhibit or promote cell elongation in the growing tip of the stem? Remember, as long as auxins are found in the growing tip, they will promote cell elongation. It promotes cell elongation, okay? Explain why the part of the plant which showed a response as illustrated in diagram B will react as shown. They want to know why was the root bending like this towards gravity. Right, and you can see, learners, here. Under the influence of gravity, the auxins tend to accumulate on the lower side of the root. High concentration on the growth on the lower side of the root will inhibit cell elongation. On the upper side of the root here, the auxin concentration is less. Therefore, cell elongation will occur on the upper side and causing the root to bend and move to grow downwards. Okay. Now, weeds are problematic to farmers because they invade farm fields and outcompete crops. This reduces the crop yield. Farmers spray their fields with chemicals known as herbicides to kill the weeds. Some weeds become resistant to herbicides. That means the weeds are so strong they can fight the herbicide and they will still survive. Scientists investigated the time it took for a species of weeds to develop resistance to 5 types of herbicides. Now you say, why are they talking about herbicides here? Remember, auxins are used as herbicides. High concentration of auxins can act as a herbicide. Herbicide means kill a particular plant. Now we don't want to kill our useful plants. We want to kill weeds. Weeds that use up the water and the light of the useful plants. So in between your grass and your mealies, you find these little plants which you don't need. They are using up the water and killing our useful food plants or food crops. And these weeds become a nuisance to farmers. So instead of using chemicals as pesticides to kill the crops, they use special kinds of auxins, plant-friendly herbicides, okay? So that they won't actually damage the actual healthy crops. So let us look at it. The results are shown in the table below. So this farmer decided to use 5 different synthetic herbicides. Herbicides made up of synthetic auxins. There's 2,4-D, Dalapon, Piclorin, Dicloflop, and Trifluralin. Right, and then they wanted to find out which of these herbicides was the best to kill a plant. And how will they know it? The faster, the more years it took to resist, it means that herbicide was a very, very effective. But if you find the time taken for weeds to develop resistance, let's look at it. 2,4-D, 9 years. Let's look at the highest. Trifluralin, it took 26 years for the weeds to fight this trifluralin. Okay? 26 years means that herbicide was very strong for that weed. The weed couldn't fight it. So it took very long to become resistant to it. Whereas this Dicloflop took 7 years. 7 years for the weed to actually fight it. So that means that this Dicloflop was a very weak herbicide. It was there to kill the weeds, but then the weeds took a very short time and then they started to fight it after 7 years. This one, after 26 years, the weeds decided to fight that herbicide. Okay. Now refer to the passage above and state how weeds act to reduce the crop yield. How do weeds act to reduce the crop yield? What do weeds do? Weeds invade the farm fields like your wheat and your mealies and your tomatoes and your potato plants. And what they do is, because they grow so fast, learners, they use up all the water and then they outcompete the crop plants for space. So that's how we need to get rid of weeds, because they'll use up the water and the light and the space and they will kill our useful plants, food plants. Identify the independent variable. What is the independent variable? The independent variable is what did that farmer do? He was able to take 5 different weed killers, 5 different herbicides. He can control, but he doesn't know how will the resistance, which one will be the highest resistant and least resistance. The resistance he does not know. How long it will take to develop resistance, he does not know. But he knows he can control a number of herbicides I'm using. So the independent variable is what he can control. It was the herbicide, okay? The type of herbicide. In this case, 5 of them, not 10. He only selected 5. So he selected it. So that's the independent variable. Dependent variable means he wanted to see how long it took for the weeds to develop resistance to the herbicide. So dependent means the time taken to develop resistance. So the more time it took, it means that that herbicide was a strong herbicide. The less time it took to fight it, that means that herbicide was a weak one. So name the herbicide to which the weeds develop resistance the fastest. They were able to, like the weeds were able to outcompete that herbicide the fastest. It was diclofloc. So this was it's only 7 years, remember. And then name the herbicide that remained effective for the longest period of time. They used it and was killing the weeds for a long time. It killed the weeds. The weeds didn't survive. And that was trifluorine. That remained effective for the longest period of time. Okay, that was trifluorine. The scientists use the same weed species when investigating resistance. Now, why do they use the same weed species for the different types of weed killers? You can't use different weeds because then you won't know, did these weeds already develop resistance? So you must use the same weed species for validity, okay? So how would the scientists have determined the resistance of the weeds to the herbicide? They would apply the herbicide to the weeds and see if the weed survives. If it survives, it means that if it survives, it means that it is not resistant, okay? But if it does not survive, it means it is resistant. Do you understand, learners? So when you resist something, you're fighting back. Okay, so they would apply the herbicide to the weed and observe if the weed survives. So now if it survives, it means it is not resistant. Okay, if it survives, it's not resistant. All right, that means if you survive it. But if you die, if the plants die, if the weeds die, it means they are resisting. Okay, we will get that right just now. Okay, learners, let us just go back 1 minute.
The road to success is to commit to yourself. In order for you to become who you want to become as a student, you need to commit to reading your books, to studying, to bettering yourself. I know it's not going to be easy. Actually, no one said it's going to be easy. But if you put in the work, the results will show in your life. Tap into your power. Sometimes you will feel like putting away your books, and not reading as much as you're supposed to be. But I want you to commit, because through commitment, there is infinite power. The road to success is to commit to yourself. In order for you to become who you want to become as a student, you need to commit to reading your books, to studying, to bettering yourself. I know it's not going to be easy. Actually, no one said it's going to be easy. But if you put in the work, the results will show in your life. Tap into your power. Sometimes you will feel like putting away your books, and not reading as much as you're supposed to be. But I want you to commit, because through commitment, there is infinite power.
Okay, learners, welcome back one more time. I just need you to know the understanding of resistance and non-resistant. If you are not resisting, right, you can't resist, you are unable to resist, it means that that weed will die because it's not resisting, it's not able to fight back. But if it's able to fight back, it will survive. It means it becomes resistant. So if you survive, the weed survives, then it becomes resistant. But the longer it takes to survive, it means that that weed killer is the best weed killer. So resistant means it's able to fight back the herbicide. If it's not resistant, it means the weed will die. Okay, explain how the same weed species improve the validity. We cannot change the plant samples all the time, and therefore we have to keep it the same for validity. See, they use the same weed species as if you're breeding other weed species, they may have already developed some resistance. Now, learners, the last part is very important, and that is to apply your investigation to a graph. So draw a bar graph to show the time taken for the evolution of resistance to the herbicide. And you can see here that you have your x-axis and your y-axis, and then your x-axis is what you have your 5 different types of herbicides which that farmer could control, and then the time taken was plotted here. That's your dependent variable. Time taken for development of resistance, you can see that your values are in brackets there, and you can see that the spaces between the bars are even, the bars are also even. So this, wherever the ticks are, you need to see where were the marks allocated. If they say a bar graph, the type must be correct. If you say the scale, you must also make sure that your values here or your intervals are correct. They're not one space is big, one is small. So that is the scale. Also, the labels of your diagram, of your graph actually must ensure that you have both the labels taken from your investigation, both your independent and dependent variable should reflect on your bar graph. So learners, this brings us to an end of the investigative questions. Remember, you need to know your independent variable is what you can control, your dependent is what you measure, and you have to also changes. Both your variables should be in your topic, and also you need to know the scale and your values, and that they must all be equal. Thank you, learners. The more you revise plant hormones and the more you go through investigative questions, you will do well. Thank you.